All right, hello everyone. My name is Michael Schatz, and I'm your host for today for the Galaxy Training Academy's Meet the Expert series. Our guest today is Dr. Aaron Quinlan. Aaron is professor and chair of human genetics at the University of Utah and a leading figure in computational genomics. His lab is well known for combining computational advances with genomics technologies to study the causes and consequences of genome mutations and understanding the genetic basis of rare diseases. He is also well known for supporting open source software for the genomics community, including several widely used tools such as BEDTools and Mosdepth, which have become foundational for analyzing genomic intervals and sequencing data at scale. To learn more, visit his lab's website at quinlanlab.org. Aaron, thank you so much for joining us today.
It's a pleasure, Mike. Thanks for having me. Really
All right,
excited
it's
about
great.
this.
It's great to be here. So, what I'd like to do today is talk about your past, present, and future in genomics and genomics data science. So, just to kind of kick things off, tell us a little bit, how did you get started? Were you always interested in genomics?
No, I was not always interested in genomics. My path is a bit circuitous, I guess. I think Mike, you and I have a similar background in the sense that we both trained in computer science, but I think my background is even more complicated before that. I was really into art and thought I was going to be an artist. I do not have actually the skills required, but thankfully, going to high school in southwest corner of Virginia, um, I had the fortune of attending a school called the Southwest Virginia Governor's School, and that was my first introduction to programming as a junior in high school. And that quickly taught me that I was more interested in using programming as a creative outlet than art. And it turned out that I thankfully had a little bit more skill and aptitude there. So I went to undergrad at the College of William and Mary, where I was a computer science major. I think at that time, it was a very small major. I'm sure it's changed now, but I think there were 7 people in my class that were majors that year. And so I graduated in 1997. And as you probably know, Mike, back then, if you could code at all coming out of undergrad, you could get jobs as software engineers for consulting companies or lots of different options. So I started my career out of undergrad as a software engineer for a company called Price Waterhouse at the time. It then became Price Waterhouse Coopers, and actually learned a tremendous amount there about software design and thinking through what your code should do and designing tests and important things like this that we talk about all about in science now for reproducibility. Um, but then, well, let me fast forward here. I got bored with that job and decided to go back to grad school. This is right around 2001. Uh, Human genome sequence had just been completed, read some lay articles about that. And I remember vividly seeing a quote in one of the articles talking about the role that David Hausler and Jim Kent had in developing the tools necessary to do genome assembly and things like UCSC Genome Browser, which we
Yeah.
take for granted now. And there was sort of a call to arms for people with programming skills to sort of launch the post-genome era. And that was enough for me. Went to grad school. Well, I applied lots of places, got into one. Thankfully, Boston College took me. I did a PhD with Gabor Marth there and did a lot of fundamental work in genomics and was just so fortunate to have a mentor that worked on tool development. And that's really what I wanted to do. And, that's where I am. That's what I'm still doing today. So it was really 2004 when I first got into genome data analysis. This is, if you remember,
But
Mike.
that's amazing. So, I mean, so you had undergraduate in computer science, you're working in industry, and you like read a report about all the things going on in the Human Genome Project. When you applied to grad school, was it when did you write about specifically about genomics, or was it more of a general computer science kind of program that you were applying to?
I I think I wrote very poorly about genomics and what one could do, and it was very much framed from an algorithmics background. I think I was talking about like lower bound complexity or something in my letter. But I don't know how it was for you applying to grad school, but at the time for me, there wasn't much interest in people who didn't have biology. I didn't have a biology degree. nor did I have research experience in biology. So I think I applied to 15 schools and got waitlisted at one and accepted at Boston College. It might have been because of the statement that I wrote, but I think it was mainly just there was less of an appetite for strictly computational people at the time. I mean, it's changed completely now. I serve on a missions committee here, and that's often exactly what we're looking for, but
Yeah.
times have changed, thankfully.
Amazing. So you mentioned, you came in with, a lot of interest in software development and you were talking about algorithmic complexity. So what were your first projects like?
Wow, that's a great question. My first rotation project was with a wonderful man named Peter Clote at Boston College who studied the protein folding problem, which, as we know, now there's a Nobel Prize associated with the algorithmics there. Back at that time, in that time, I think the state of the art was like 70% accuracy on a very limited training set, like predicting structure from sequence. And my first project was to develop a support vector machine application to protein folding. In hindsight, I have no idea why the mentor thought that that would be the right solution, nonetheless. But my first project with Gabor Marth was this is still at the time when Sanger sequencing was the primary data source. So I ended up, it gets to open source, actually ended up working with the Fred and Frat programs from Phil Green's lab and hacking that C code to try and call heterozygotes from Sanger sequencing data. That was the early first phase of data coming out of the ENCODE project, and worked vigorously on that for about a year, only to be scooped by Matt Stevens and Debbie Nickerson, who basically did the exact same thing but better. Um, so that was my very first project. A bit let the wind out of the sails a bit, but then Illumina then Solexa sequencing and 454 had just come out, so we had a really nice, fun, exciting collaboration with the WashU Genome Center to develop software for variant calling and alignment and base calling. And that's the first tool I ever wrote was a closed source tool called Pyrobase. And the 9 people that used it around the world, I think really liked it. But I learned I learned a lot in doing that.
Well,
Um,
I'm curious, what was the rationale behind closed source versus open source?
Yeah, it's a great question. I sort of teed that up so I get the question. Um, the mindset in Gabor's lab was there just wasn't an understanding of the value
Hmm.
of open source. And I think this gets at sort of the transition that's occurred over the last 20 years in terms of mindset about software and science. He had developed a very beautiful piece of software called Polybase, which was the first tool to use Bayesian logic to emit posterior probabilities for variance. And the lab that he did his training in was very much focused on closed source stuff. And so that was the mindset he came into his lab, that he brought to his lab. And I'm not saying it as a criticism to him, it was just the culture.
Yeah, yeah.
But yeah, actually, um, talking to James Taylor and others at the time when I was developing software when I started my postdoc changed my mindset completely.
Hmm.
We can talk about that more if you'd like, but
Yeah,
yeah,
actually, I'd
started
be
out.
very curious to hear from you. What were those interactions with James like?
Yeah, so I think I first met James when I was a postdoc in Ira Hall's lab at UVA, and this was around the time that I had the initial thoughts for developing bed tools. And if I remember correctly, I think this is mostly right. These memories are a bit vague. It's like 2008. But I reached out to him by email. He didn't know me.
Yeah.
I reached out to him by email and asked if I could get some guidance about how to develop software for open source and what that meant. Because I knew of this is the early days for Galaxy.
Yeah.
I knew of him. I think he had just had been a couple years in his job at Emory and was very gracious and offered to get on a call like this back then. I guess it must have been Skype or something. We just talked about, well, he really encouraged me to make it open source so that I think what he realized really quickly early on is just once you do that, you have a team of developers and you have a team of people supporting something, and obviously a team is going to make something far better than an individual. And I understood that. That I really embraced that quickly. I think the other piece that I didn't know how to do at the time for my software engineering back in those days, we used very old school version control technology. So just like learning about Git and other technologies like that to even manage the software, he was incredibly helpful. And then he actually pointed me to some of his open source code related to genome arithmetic um, from BEDTools. I forget what it's called, BX Python. I forget what he called that library, but it was kind of like the original, some of the original genome arithmetic stuff in Galaxy, and we kind of riffed on that. Anyway, it just kind of opened up my world in terms of how to think about designing software and sharing it with the community because I understood the concept but didn't really know the practical details.
That's amazing. I mean, I'd say BEDTools has emerged as like one of the most popular, important software tools in bioinformatics. I mean, did you know it'd become so widely used? How, like, what kept you going on it?
Oh gosh, no. Yeah, the I have to give Ira Hall a lot of credit for this. I so my postdoc with him was to study structural variation in different laboratory mouse strains. And I had developed a different piece of software called Hydra to find structural variants with paired end Illumina sequencing. And that was really fun. But then we launched into this much more challenging set of experiments, which was what are these structural variants doing?
Yeah.
What do they, which impact, just basic stuff like does it affect a gene? How many per chromosome? Where are they? What's their size? What's their spatial relationship? All these kind of fundamental things that we take for granted now. And the only tools I knew of at the time that could do that were coming out of Jim Kent's uh, code, that underlying UCSC. I forget what those tools were called now, but I struggled to use them. They did some things, but not everything I wanted to do. James had some tools in Galaxy that did some of this as well. Um, but I started dabbling as a side project and kind of framing out concepts. So I did a lot of design, a lot of drawings, and just laying out the different types of things that we would want to do. Um, because what I realized I was doing over and over in Iris Lab was just writing custom scripts to do stuff. And um, anyway, fast forward to a couple months in, I asked Ira if he would be okay if I took a couple weeks and just focused just strictly on BEDTools and writing a first version. And I was really nervous about the conversation because he was an early career assistant professor trying to get tenure, and we were on the brink of a paper. And to his credit, he was like, "Go for it." And I did that, and then we wrote, maybe a 500 word app note about it, kind of expecting that might be useful to some people. And I had just put it up on what was the predecessor to GitHub that everyone used? SourceForge.
SourceForge. Yeah, sure.
SourceForge. And very quickly, and thankfully, like you, I knew some people from going to meetings. I knew other postdocs,
Yeah.
so shared it with some people. You might have been one of them. And very quickly, people started using it, and it really surprised me. I knew it was generally useful, but I didn't really appreciate how many other people were struggling with the same issues. But then once I moved it to GitHub, which was much easier for a community to use, uh, there's hundreds and hundreds of contributors now, which has just been a real joy to see.
Yeah, that's amazing. Tapping into the broader community and supporting them while they support you. That's a virtuous cycle.
Yeah, absolutely.
In my own work, it feels like in almost every paper, maybe there's some preliminary analysis, but then there's a section where it's like, oh, and then we use bed tools for this overlap and this enrichment and this test and this test.
Hmm.
And within Galaxy, within our training academy this week, many of the tutorials use bed tools. I guess I'm wondering if you have any tips or suggestions to the users on how to think about it or how to best use it.
Ooh, that's a great question. Yeah, I mean, I guess the way I the way I use bed tools, and the way I encourage people in my lab to is Um, I think certainly the collection of tools can do almost anything with genome annotations or genome intervals. I think the what I found for people that are new to it is it helps to kind of draw out what you want to do with the data, what you're looking for, sort of think about your expected results. Because I think if you use them for a while, it becomes kind of intuitive of how you chain the
Yeah,
output of one tool to another. But I think initially it can be a little challenging. So this is part of the motivation for the documentation of drawing out what
yeah,
actually happens with each of the operations, and that's what I would recommend if people are struggling with thinking creatively about how to use them. I spend time with a pencil and a piece of paper. I think it really helps you to lay out first draw what the expected result is, and I think you can work backwards from that to iterate on the results. Because like anything, you need positive and negative controls, and you need to be able to verify that the results are right. Um, yeah, that's what I teach in my course I just finished this year, actually, or this week.
excellent. Excellent. Yeah, I use those same training materials, those little diagrams of how the intervals, because sometimes, there's a lot of sort of parallels to, I don't know, set logic from SQL of like the left
Sure.
join versus the inner join, outer join, all this. So it's really useful to see all those diagrams.
Yeah.
One
And
thing I
and
would ask is there anything you'd wish you had done differently in its design? And I think I did notice that you have a new project to kind of reinvent bed tools.
oh my gosh, so many things. But it's yeah, like everybody, I've been following, the advances in coding agents, and I've actually thought about rewriting bed tools from scratch with the help of coding agents to deal with. I mean, it's a huge code base, so it's a massive undertaking that I no longer have time for. Um, but yeah, so I think one of the biggest challenges with the code base is that um, it's not well designed for flexibility in the sense that each tool gives a very specific output given the inputs, but often users want to. I think a great example is you intersect two files. It's going to give you the list of intervals that are shared between those files. But often what people want to do is, well, if the output meets this criteria, I want to do this type of thing. If it
Hmm.
meets a different type of criteria, I want to do a different thing to it. So essentially conditional logic or filtering on the output. And
Hmm.
that's what is really, really difficult with the design embed tools to allow users to have that type of flexibility. So that's the motivation behind this other project that we've written called Better, BEDDER. So it's a Rust library that is intentionally abstracted so that we can allow people to write simple Python functions that you pass into the code so you can filter and manipulate the output record by record rather than bed tools just gives you a batch of output and you
Hmm.
have to do things with it. So yeah, like any project, a lot of regrets about the design, and it's an old C++ library, and I think if it were written today, I'd probably do it in Rust because that's the modern language, and I think it's a much safer language. But I'm old and I'm not very skilled in Rust, so I can't do that.
I mean, what you described, I mean, if you talk about old, what you described feels a little bit like awk, right? Where you could kind of stream through a file and then every line you could have all kinds of conditional logics.
That's the yeah.
I guess I'm wondering, where that inspiration came. Maybe it was awk, but where that inspiration came from.
Um, I think yeah, Awk might be part of it. So you probably know, I've for a long time been collaborating with this incredibly talented developer named Brent Pedersen, and he and I have written well. So getting back to open source, BedTools was the first open source tool I've written, and I guess my lab has published like 60 open source tools at this point. And Brent's been a part of probably 20 of those
Wow.
over the last 10 years. And um, yeah, I think he and I spent a lot of time. So Better is funded by a grant from the NHGRI. And he and I spent probably the first 2 years of that grant just thinking about design. not only just like core algorithms, but just like what interface would best empower users to address the hundreds of issues that we've encountered with bed tools along the way. And I really still to this day love the basic Unix concept of one tool to want to do one thing very simply, and then you develop your fancy Rube Goldberg machine by chaining all those things together. And I think awk is a perfect example of that. It's a very powerful tool. Uh, I'd say awk that the interface that we have in Better is very much inspired by awk. But I think with Brent's um, special sauce woven in, so to speak, he's an incredible developer and just a very creative scientist.
Excellent, excellent. Well, I mean, you just mentioned what a big component open source software is in your lab, but I know you're also, deeply involved in a number of like really pioneering human genetic studies looking at, I don't know, transmission of de novo variation and somatic variation. Is there anything that you're particularly excited about now?
It's been, thanks for bringing that up. I mean, thinking about what my lab works on now, 11 years after moving to University of Utah versus what it worked on when I moved here, it's changed quite a bit. I actually think of myself more as I guess, a geneticist at this point than a computational biologist. I also chair a genetics department out here, so I have to think more broadly about genetics and connections with other translational contexts. We're in a school of medicine. Um, so one of the things that I am extremely excited about is, as you probably know, Utah is pretty famous for these very large multi-generational pedigrees that were part of establishing the linkage map for the Human Genome Project and things like that, these so-called Ceph Utah pedigrees. One of my close colleagues, Deb Necklason, starting in 2000, started recruiting the fourth generation of these pedigrees, expanding them, and we did additional really deep phenotyping on all these individuals. So everything from like, do they have perfect pitch or not? What's their hearing? It's insane how much phenotyping there is. And so I think one of the really unique things that we have the opportunity to do here, I guess perhaps much like what Decode in Iceland with the families they have there, we have a very unique ability to study mutation, which is the core biological question in my lab, causes and consequences of mutation, and its connection to phenotypes. So one of the things I'm very excited about, we're in the middle of a study where we've sequenced all four generations of these families, and we're trying to understand how variable mutation rates are from family to family, and what are the genetic and environmental factors that modulate both mutation in sperm and egg and in somatic cells in individuals, and then following up on some really interesting connections that we've observed in the past between mutation rate and outcomes, health outcomes in these individuals. So 5 years ago, we published a paper that I think 8 people in the world have read, that shows that men and women with the highest mutation rates in their germline live roughly 5 fewer years than men and women with the lowest germline mutation rates. And the hypothesis is that those individuals also have higher mutation rates in their soma, and so their tissues are senescing at a greater rate and accumulating mutations at a greater rate. And so what we're working on now is connecting those mutation rate observations with other outcomes that we have in our electronic health record and in something called the Utah Population Database. So
Hmm.
got a team of really talented postdocs working on this, and I'm hoping to demonstrate deeper connections between one's mutation rate and health outcomes. And I think
Do you
it's
know,
real.
or could you speculate as to what the causes of that is? It, I don't know, something in the DNA replication or repair
Yeah,
pathways?
it's a great question. Yeah, so the working hypothesis, and this is not just us that are thinking about this.
Yeah.
Folks like Kelly Harris, she's really thinking about this. Molly Shaborsky is thinking about this. But yeah, so there's a couple hundred proteins that are involved in repairing damage, DNA damage. So the mutations we observe are really just the residual of damage that has occurred versus damage that has been repaired. Anything that's
Yeah.
unrepaired is mutation. Um, so if there's a couple hundred proteins that are involved in that repair, we know of TP53 and BRCA1 and things
Sure.
involved in sort of either shutting a cell down or repairing damage, respectively. Um, there's polymorphism in the human population in those proteins. So it's subtle variation, but that subtle variation modulates the capacity to repair that damage and thus should lead to variation in the burden of mutation. So yeah, the working hypothesis is the reason we observe variability from family to family in mutation is because there's polymorphism in the capacity to repair damage.
Wow.
And we've shown that, our lab has shown that so-called mutator alleles in mouse, but it's much more difficult to find and characterize them with statistical significance in humans. And that's where I think these big human pedigrees offer a lot of power because we
Yeah.
have lots of offspring and generational effects.
You mentioned the fourth generation was recruited some 20 years ago. Are you looking to the fifth or beyond generations?
Oh, sorry, I misspoke. It started in 2020,
Ah, okay.
so 5 years ago. But yes, that's part of my job as a department chair is to try to get the state legislature involved in not only expanding to fifth and so forth generations, but we want to expand the recruitment of other families
Ah. Yeah,
because
it'd be an amazing resource.
it's just, yeah, I agree. You should write her a letter of support then. I agree. It's just a really expensive endeavor.
Yeah.
That recruitment and the deep phenotyping, that was a couple $1 million project in and of itself. You got to bring people into clinic, you got to pay for their travel, all that kind of stuff. So, yeah.
Where do you see the field moving over the next 20 years? Do you ever think there'll be a day when everyone has their genome sequenced?
That's a good question. You kind of have to imagine that if not everybody, most people will. I mean, just the way that support through insurance is moving. I think the recognition that there's immense, you know, these really powerful AI models are so starved for data. Innovation comes through really large scale heterogeneous
Yeah.
data. Right? I can't imagine that there's not a strong scientific argument for doing that. So I think where I'd really like to see the field advance over the next 20 years is just the fundamental question in genetics, a deeper understanding of the connection between genotype and phenotype, because there's not much of an understanding. I mean, we know a lot about, clear loss of function variants in Mendelian disorders, for example. But I think one of the things that I think about all the time, because the other hat I wear is in our rare disease program here, with the state of the art technology, long read sequencing, getting methylation through the kitchen sink in terms of molecular techniques and computational techniques, we still can only solve maybe 30% of kids with clear genetic and developmental disorders. Why can't we solve the other 70%? What is going on? And I feel like if we can understand that, we'll have a big step forward in moving to understanding and treatment of more complex common disorders. But I think it all starts with really understanding genotype to phenotype deeply in the most extreme cases like rare disease.
If you had to speculate, where do you see the biggest opportunities to solve those extra 70%?
I think that many of these unsolved cases are for two primary reasons. One, well, for a long time, the speculation has been, well, the causal variant is in their DNA. We're just not finding it with the technologies we have. I don't believe that at all now, especially with T to T assemblies and
Where
full
could it be?
molecular
Yeah.
tests. We're finding everything that's there.
Yeah.
We don't know how to do is one, there are variants in there that we don't know how to label them as causal. We just don't know that they're deleterious. That's, I think, probably the bulk of the scenarios. I think the other one, though, is a lot of these phenotypes are caused by multiple effects in multiple genes,
Yeah.
leads to a phenotype that looks like something that we would call condition A. And we're just not, we're our understanding, we don't have good models for how to identify those types of situations. And
I completely agree with you, right? You know, we've learned a lot by looking at sort of one variant at a time, but I think we're kind of running out of the list there, and we need to be looking at sort of combinations of variants and the epistatic interactions. That kind of has to be. I mean, if you think about, I mean, it's a crude analogy, but I think it's a useful analogy. If you think about the phenotype of a software program, right? Where you could have, changes in the logic in many different positions, changes of many if statements could lead to sort of similar behaviors, so we have to start developing those models that go beyond one variant at a time.
exactly. I mean, resources like Nomad have been incredible, but
Yeah.
people still use them to filter, to look at one variant at a time. And I mean, the problem is the combinatorics of this is just it's astronomical. And so I know folks like Caitlin Smoha are thinking about combinations of variants within one gene, and
Yeah.
that alone is hard in Nomad. But now you start thinking about all the combinatorics of
Yeah.
multiple haplotypes for multiple genes. But yeah, so I guess to your question, I don't know if it was where would you want the field to be or where do you think it will be? Well, where I would want it to be is that an understanding in the next 20 years of what's going on in those situations, because I think it'll lead to much deeper understanding of all sorts of phenotypes and probably empower treatments in ways that we can't even appreciate now.
You've kind of hinted at this, but uh, in sort of the spectrum between innovative genomics technologies to innovative computational technologies, do you see, will it be a combination? Will it be one leading over the other?
I think it's definitely a combination, the relative balance. I'm not sure, but it's a great question. It's a timely question. It's something I actually gave a talk about at my university on Friday about our core infrastructure. So bear with me here. But so unlike a lot of places, we don't have a genome center here. We have some core
Hmm.
facilities for DNA sequencing. Um, and so DNA sequencing in my mind is a commodity now. I mean, you can send it out to companies
Yeah.
and I get it. There's a lot of rationale to not have a genome center institution. But I think where innovation really comes these days is by tweaking these technologies to be at the cutting edge of really clever um, molecular techniques. I'm thinking of, for instance, duplex sequencing, where you get really high accuracy
Yeah.
resolution on mutations. That requires a lot of iteration and feedback on the technique, and simultaneously using the molecular output to train models to figure out, to adjudicate what's like a real mutation, what's not. Even though those technologies lower error rate by orders of magnitude, there's still errors. You still need to be able to tease out signal to noise. And so I think with all advances in molecular technologies, there needs to be commensurate advances in the computational technologies. I think it's the two in kind that really drive discovery. And I expect it will stay that way forever. I mean, genomics is fundamentally data science.
I agree. I mean, you mentioned, I don't know, one of your first projects was support vector machine. You mentioned a little bit about other models. Like, what do you see as the future of AI in genomics more broadly?
Phew. Yeah, I'm on the periphery of expertise there, but I know one thing that we are really excited about in the near term future that is difficult here. I don't know how it is at Hawkins is um, we don't have the ability currently to use things like Claude and other tools in a PHI
Uh,
environment. We really want to integrate electronic health record with genomics and imaging and all these things. To, maybe fork existing models, but I think even more fundamentally to use, for your advanced scientists that understand code and want time savers, to use those technologies to deploy projects and ideas in a protected and a PHI environment. Especially as NIST standards and NIH standards for regulated computing environments become ever more onerous. I think if we don't solve that and develop solutions to deploy AI models in a research context, it's going to be kind of two different worlds. It's going to be huge advances in non-human contexts, and anything related to human data is going to be left behind unless we figure out how to deploy these technologies in the forthcoming regulated environments. And that's something I'm really pushing on here because we all know, I mean, you don't have to be Mike Schatz now to write awesome code. You just have to have a really good idea and spend a
Yeah.
few hours with Claude Code, and you've got very nice piece of software that can execute the idea that you had.
One thing, just to give a little teaser, we're very actively working on is making those integrations into Galaxy very deep, where you can kind of, program it interactively. And then furthermore, we're in a place now where we can run some of the genomics models, like Alpha Genome, in a protected way inside of Galaxy. So we'd be thrilled to
Really?
talk to you more about this.
Yeah, I would love to learn more about that. That's exciting.
Yeah.
That's
But I agree with you. I mean, this is clearly like the direction the field is moving, and we're here to try to support that very deeply.
Yeah, and I would say just in terms of our, advocacy going forward, I think it's folks like you and me and others who have whose voice can be heard in this area. I think the other piece is advocacy with the
Yeah.
NIH about how some of these policies are really going to make science as we know it, computational science as we know it, very, very difficult.
Yeah, I agree. There's been some statements from NIH, but they're a little bit ambiguous, and there's a lot of nuance here. Some people interpret it, you can never use AI models, but the reality is that there isn't a role for them. There is an opportunity for them. You just have to be very careful because the models, if you're not careful, can leak even in unexpected ways.
Yeah.
I'm very excited about them.
Yeah, and that's the obvious concern for academic medical centers. The leak is not no good.
Correct. 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 Well, thank you so much for your time. I have one sort of final question for you
Sure.
here.
Yeah.
We're in this training academy that has, some experienced people, but also a lot of beginners. I guess I'm curious if you have any advice for students that are entering the field, but they're not really sure how to get started. Like, what's the path forward for them? What should they be paying attention to?
Yeah. That's a great question. I think so. I have recent experience with this. I have a very talented new graduate student who comes in to the lab and to the department with very little background. And the thing that we've talked a lot about is something that I didn't learn until probably too late, 8, 10 years in. Really understanding probability and
Mm,
statistics. I mean, and just thinking about how coding has advanced with AI agents, I think obviously you need to make sure you understand the fundamentals of programming before you use tools like that so you can vet them. But I think ever more we're getting into research contexts where what really matters is your ability to model and understand the sources of noise and bias in your data so that you can actually understand what's true biology and what's junk. And I guess every project that in my lab now, it fundamentally our decision to publish the result comes down to that.
mm,
Is really what is the appropriate um, model of the noise, and we spend a lot of time thinking about that. And how does that potentially explain what we perceive to be biology? So to get back to
yeah.
your question, I would really invest in, there's amazing resources now, both in terms of YouTube videos, but also just partnering up with AI agents to really do what Feynman did, keep a list of all the things that you don't understand about probability and statistics and just work and work and work to understand them. Because I think once you have some capacity there, it really liberates you to think creatively about projects and really head off problems and analysis. And I think it expedites projects. So if I were telling myself as a first year grad student to focus, what to focus on, it would be that because I didn't really focus on it until late in my postdoc.
I love that sentiment. I've come to be at peace with the fact that my intuition for statistics is pretty weak, but I will often write like a little simulator. You can throw a bunch of balls into bins and
Yeah,
empirically
exactly.
look at what happens if you change this probability or that probability. That's served me tremendously to like have those
Simulation.
simulators that helps me develop that intuition. So,
Yeah, I don't have the math background to come up with the analytic solution, but I can write the code to do the
yep.
simulation.
Yeah, exactly. I think that's outstanding advice. Well, Aaron, thank you so much for your time and attention. We're really excited to learn more about better in the, I don't know, coming months and years here. That sounds like a really important advance. And then also, your work in genetics is outstanding and amazing, and I look forward to seeing those papers in the near future. So thank you very much for your time.
Thanks so much, Mike. Really appreciate it. Glad to be here.
Okay, good luck to everyone on the Academy this week. See you soon.